Okay, hello everyone. It couldn't be as emotional, I'm sorry. Good, so I'm in my lab we study real evolution, biological evolution, but as you know, I have a passion for science, so let me try to adapt what I'm discussing today in analogy to language, something that I would like to call genetic onomatopoeia. If it's not really onomatopoeia, don't take it too seriously. This is a question for the non-Hebrew speakers among you. One of those objects in Hebrew, or in Israeli, is called kos, and the other is called bakbuk. Can you guess which is which? I don't need to be Victoria Hanna to pronounce bakbuk to give you a huge hint that this is probably the bakbuk and this is the kos, right? But if I ask you one of these objects is called kiseh and the other shulchan, in principle, you shouldn't be able to tell me which is which. Right? There's nothing table-ish in a table, there's nothing chairish in a chair. In principle, you cannot know. And if we use the term of signified and signifier, the signified is the object, and the signifier is the name that is given to it. Excuse me for using lousy linguistic terms. We know from the Saussure that the connection between them, in the majority of the cases, onomatopoeias are just exceptions, beautiful, but exceptions, the connection between them is totally arbitrary. Namely, we could have called kiseh shulchan and shulchan kiseh. We could have agreed that a chair is a table and a table is a chair, and everything would work. It wouldn't make much sense to call this bakbuk and this kos, but otherwise, in the majority of the words in any language, onomatopoeias are rare, and the arbitrariness of assignment within the signified and the signifier is the norm rather than the exception. Okay? What about the genetic code? Can I use this analogy just to convey to you something that we have discovered in the lab? So in biology, at the heart of genetics, we also have two systems of chemicals that I would like to refer to one of them as the signifier and the other one as the signified, and this is the heart of the genetic code. So let me teach you about the genetic code in a few minutes. So the DNA and the RNA, you heard about RNA from Adi, the viruses that have RNAs, their genomes. The DNA, the RNA is a language written over an alphabet of just 4 characters. These are the famous A, C, G, and T. In RNA, it's U instead of a T. It doesn't make much of a difference. It encodes for amino acids, which come in an alphabet of 20, and since 4 cannot encode uniquely 20, there is a system of codons, triplets of nucleotides, that uniquely define each amino acid. So, for example, this amino acid up there is consisting of a sequence of 3 uracil nucleotides, etc. And as you can see immediately, it seems that the genetic code obeys the arbitrariness because there is no simple connection between the chemistry of the nucleotides and the chemistry of the amino acid encoded. In fact, in this example, this bulky amino acid, this big amino acid, is encoded by small nucleotides, and this small amino acid is encoded by large nucleotides, suggesting that at least in terms of the bulkiness, there is a totally arbitrary assignment between the codons and the amino acids, between the nucleotides and the amino acids. And the structure of the genetic code conveys that notion fully. There is an arbitrary assignment, as long as it's agreed upon in organisms, that conveys or that maps triplets of nucleotides into amino acids. Let me tell you a little bit about the structure of the genetic code. How do we read it? Again, it's triplets that encode for amino acids. So, for example, the first amino acid, phenylalanine, is encoded with a U at the first position, U at the second position, and U at the third position, although C at the third position would also encode for that same amino acid. So in that respect, U, and C are synonymous, but the code is arbitrary in the sense that, in principle, any combination could have encoded for any amino acid. In that respect, it reminds me of the Morse code that only has 2 types of characters, a dot and a dash, as opposed to 4 in DNA and RNA, and the Morse code is arbitrary in the same sense, because if you look at E that only has a dot, it's there's nothing dotty or dottish or dotty whatever in an E. It was just agreed that this is how you encode for E. And the same goes for the rest. This is not to say that this code or these codes are random, not at all. The genetic code is, we believe, is a result of very profound evolutionary selection. It's an excellent code, much better than many arbitrary code, as much as the Morse code is an excellent one. For example, the most frequent signs in the language are characterized by short encoding, and similar characters have similar meanings, or mathematical quantities have similar encoding. So it's an excellent code in that respect. And the same goes for the genetic code, but it's arbitrary in the sense that there is no absolute connection between the signify and the signified. Now, let us take this knowledge to tell you something that we have recently discovered in the lab. This is an artistic view of a cell. Cells don't really look like that, but this is just to convey the notion that cells are a very complex entity. They are very crowded, and on top of that, molecules appear to have addresses, destinations within the cell. And Orna Choder is here in the audience. She was actually the first to have brought that extreme order to my attention, to the attention of many others. Molecules in cells have addresses. They don't just float within the cells. How does the cell know how to destine each molecule within the cell? There must be a language, a zip code, in fact, that would be embedded within the proteins and the mRNAs that would tell the cell where to put each one of them. Let me give you a more simplistic view of the structure of a cell. And this is a cell here that has a DNA. It has RNA, which is a transcript that goes to the cytoplasm where it's translated into proteins. And I want you to refer in particular to three types of proteins that cell makes. There's the proteins that are inside the cell. There are proteins that are secreted from cells. They are going into the medium. If it's a unicellular organism, they help build communities, public good, or weapons. And there are proteins that reside within the membranes of cells. So three types of cells. And the question is, how does the cell know the zip code for each of them? And for many years, the zip code was thought, and still is, known to reside at the level of the protein itself. There is information in the protein that doesn't only specify the function of the protein, but specifies where within the cell it should be located. But we thought, could there be some sense of genetic onomatopoeia that would allow the cell, already before translation of the protein, where there is only RNA, would allow the cell to say, this RNA is, when translated, it's going to be a protein that is secreted outside, and that RNA, when translated, is going to be a protein that is embedded in the membrane, and the third RNA, when translated, is actually going to reside within the cell. So if that were possible, it would have amounted to some onomatopoeia structure, perhaps resembling the book more than the table. But the question is there any reason to believe that there is such a code, that there is any such onomatopoeic structure? So for the biologists among you, you could, of course, ask me immediately, what are you talking about? We know about the signal peptides. Signal peptides are sequences embedded in the translated amino acid that tells the protein if it needs to be translated and if it needs to be, or if it needs to be embedded in the membrane. But we know that the signal peptide model is incomplete in the sense that many mRNAs that encode for secreted and for membranal proteins, they find their destination close to the membrane before they have been translated. And their translation, even in situ translation, does not necessarily depend always on that machinery that recognizes the amino acid sequence. So we asked ourselves, is there a code that perhaps resembles a genetic onomatopoeia? It would be embedded at the mRNA. It would allow the cell to tell ahead of time the destination of a protein. So let me tell you about one important property of amino acids. And that's the property, a chemical property called hydrophobicity. Hydrophobicity, as you know, is the tendency to hate water. And in fact, since the membranes of cells are made of lipids, if you hate water, you love membrane. If you love water, you hate membranes, and vice versa. So one can measure for the 20 amino acids this tendency of them to hate water or to love membrane, and you can sort them. These are the amino acids that are hydrophobic. They like to be within the membrane. These are the hydrophilic. They don't like to be the membrane. And let me superimpose now the genetic code on top and suggest to you, and this is partially based on an early observation that was made by Eitan Bibi and Chaim Prilusky here at the Weizmann. Some of it is actually known to people from even before that. An interesting observation: if you look at the genetic code and you color the amino acids based on this chemical property, whether they like or hate water, you see something that is hard to ignore, and this is that the first half of the left half of the genetic table is mainly characterized by hydrophobic amino acids, and the right one is mainly characterized by hydrophilic amino acids. That means that there is, perhaps, if I may use this analogy, a non-random assignment between the chemistry of the signified and the signifier. Perhaps to the extent that, and let me just mention that what characterizes the first two columns here, the U and the C, is that they belong to a family of nucleotides that are called pyrimidines, whose nickname is Y, as opposed to A and G, who are purines, whose short name is R. So this means that a codon that encodes for a hydrophobic amino acid takes the form of NYN, N being any nucleotide, because you can be wherever you want in the first and in the first base. Y specifies that it needs to be either U or C, which is this combination, and then it's followed by any other nucleotide. So if you want to encode for hydrophobic amino acid, usually a Y in the middle would ensure that, usually. Now, remember also another property, and it's important for the rest of this journey, that the code is redundant in the sense that if you have specified the first and second nucleotide within a codon, the third is typically free to mutate. Namely, synonymous codons would be different in the third position of the codon, and they would still encode for the same amino acid. So these are two important properties of codons. Hydrophobic amino acid would be NYN, and the third position is going to be relatively flexible in evolution. So the question is, could cells use that regularity, that interesting non-random assignment between the chemical properties of the codons and the amino acids to generate a zip coding system that would not be dependent on translation of the protein? So let me tell you something about membrane proteins. This is a segment of a membrane of a cell, of a hypothetical cell. And as you see, there are many proteins that traverse the membrane. And if you look at this particular one, it has typically 20 amino acids in a row. This is what it takes to traverse the membrane. And since the membrane is hydrophobic, it's made of fatty acids, you need hydrophobic amino acids in order to reside that. It's like, oil likes to be with oil, never with water. So we realized that a sequence, the nucleotide sequence, not the amino acid sequence, the nucleotide sequence of a segment that goes through the membrane should look like many repetitions, perhaps 20, of the form NYN, 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 etc., perhaps uninterrupted. Okay? So could that serve as a way to destine the mRNA for a membrane protein to the vicinity of the membrane before it was even translated? If so, that could function as a zip code that resides at the RNA, and it predicts ahead of time the result of translation without the need to translate in reality. Okay, but then we thought, what about secreted proteins? Secreted proteins, on one hand, they don't have hydrophobic amino acids. On the other hand, their mRNAs would better be translated in the vicinity of the membrane. That would facilitate their secretion to the external milieu of the cell. So there is a problem. On one hand, it would have been nice if they had this pattern. On the other hand, there's no reason for them to have the pattern because they don't have hydrophobic amino acids. But then we realized if we just ignore all the spaces in this long sentence and rebuild it again, putting the spaces with one shift to the right or to the left, we are getting something that secreted proteins could, in principle, use as their zip code at the mRNA level, despite the fact that they are not made of hydrophobic amino acid. And why is it? If they have codons of the NNY form rather than the NYN form, from the point of view of an entity, hypothetical at the moment, that reads the text without awareness to the gaps, it looks just the same. Because what's common to both of them is that you have pyrimidines, this Y, in jumps of 3. So we said, perhaps originally a system has evolved that would take that NYN repetitions as a zip code to localize mRNAs from membrane proteins, but secreted proteins could have co-opted that system and use the repetitions of Y in jumps of 3, but with an offset such that utilizing, in fact, the redundancy of the genetic code. Because if you remember, I told you that on the third position of the codon, you can essentially put almost always any nucleotide that you want. And if a Y is needed, this is exactly the place to stack it, evolutionarily speaking. So we've examined that. First, bioinformatically, and when I say we, I mean in collaboration with Jeff Gerst's lab in our department, with Cohen-Zontag as the first author, and indeed it turns out to be the case. Indeed, it turns out that membrane proteins feature uninterrupted runs of NYN, 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 whereas secreted proteins tend to feature runs that are also uninterrupted of pyrimidines of Ys in jumps of 3, but in an offset of 1 nucleotide. In fact, when you do the statistics, this is exactly what you are getting. I'm not getting into the details, but I'll just tell you that we have now a new regulatory motif that is present. It's a word that tends to repeat itself within the mRNAs that encode both for membranal proteins and for secreted proteins, and it has several interesting features. For example, it indeed features uninterrupted runs, 10, 15, sometimes even more, of pyrimidines in jumps of 3. This is very highly unusual. At random, with proper randomizations, etc., you wouldn't get it as much as you see it in reality in secreted proteins. It has additional interesting properties for the biologist among you. It can appear in the UTRs as long as you have it. This is a localization signal, we believe, and indeed we have shown that increasing or decreasing the content of these pyrimidines in the mRNAs of proteins that are encoding for secreted proteins can have an effect on their production and secretion, etc. So now I want to tell you towards the end how we utilize this interesting motif that could not have been, by the way, discovered by the regular motif finding algorithms. It's a very elusive motif otherwise. It was only through the inspection of the genetic code, perhaps this analogy to the onomatopoeias that allow it to be discovered. But now we want to take it to answer a very important question in evolution of cooperation. And a lot of what I know about evolution of cooperation is from Martin Nowak, who gave a beautiful talk in Ilanit. I hope many of you heard it last week about the difficulty to evolve reliable cooperation. Why is it difficult to evolve cooperation? Because the tendency, as Adi Stern showed us in the previous session, the tendency to, or the temptation to defect is always there. And we are looking at a particular cooperation. So this motif that I told you about is present in many of the public good proteins that yeast cells and other cells are producing as a way to form their communities. And one such protein is called SAC2. It's present in a yeast that we love to study. And what SAC2 is a secreted protein. It's symbolized here with this X here. And what it does is that it cleaves a source of food that is otherwise indigestible. So now imagine a community of yeast cells. They live together and they secrete that protein as a public good. This is a very communal thing to do. Each of the participants secretes something, and then you have a functional society. Everyone contributes the effort, and everyone can enjoy that. We want to ask the question, to what extent this motif that we discovered that tells the cell to what extent to secrete or to donate some of its resources to the community, to what extent this is actually governed genetically by the motif that we have discovered. And this reminds us, of course, of the classical prisoner's dilemma in which the temptation to defect upon others is very large. And in fact, many of the challenges in understanding how evolution has emerged is to understand how it was encoded genetically in such a stable way within biological systems. So what we thought was that perhaps the syntax of this motif encodes a semantics that governs the decision within such a prisoner's iterated, perhaps prisoner's dilemma game, whether to cooperate or not. Now that we have a knob rather than a switch, defect or cooperate, a simple-minded dichotomous option, perhaps evolution could use that by adapting the structure of this motif, the syntax of this motif, in order to determine what would be an evolutionarily stable strategy of secretion versus retention for each of the public good proteins that a cell does. So let me just say very briefly that what we do, and when I say we, it's my two very talented students, Ruti and Vir, who are, I think, here in the audience. What we do is that we actually engineer yeast cells and change the syntax, and we hope also the semantics of these motifs for this particular gene that encodes for a public good protein, and determine whether they would secrete more or less, how generous would they be, but again, not in a dichotomous fashion, but as an analog knob. And we co-culture now different variants of this motif in order to perhaps get insights into the evolution of this motif. How did it evolve over time? There's much to say about it, and even much more Dvir and Ruthie tells me from result from just a few days ago, but I think I will rush quickly to the questions. And before that, just the acknowledgements. These are the people who have contributed. I will not read their names. And just at the very end, towards your questions, this resource that Josh and Martin and many other people are using is really marvelous. If you go to Ngram Viewer, you can type in any word, and you would see how frequently it was used in many different languages. So Einstein, Frankenstein, Sherlock Holmes, etc. At some point, after playing a lot with that, I wanted to see what happened to punctuation marks. And in particular, I looked at the question and exclamation mark, and I had the intuition to pick up German. And this is what I saw. And this is just to motivate the question session here. So this is the usage of question mark and exclamation mark in German literature over the last 200 years. And we see a trend that is tantalizing, I think. We start 200 years with questions being 2 times more frequent than exclamations. Then the gap gradually narrows down. Alice would have been much more popular than question mark in these times. And it closes, and it once just around the First World War, and then it opens again, but it closes around the Second World War, and then it reopens again. By the way, the same trend, very similar trend, is observed in Italian. No phylogenetic, although historic relationship is obvious. It is not observed in English, in which the gap has safely been constant. So I think this is interesting as a way, perhaps, to understand evolution of even consciousness of people, and also to motivate you to ask me questions. So thank you very much.
So questions?
So the question was, in the table that I showed you, there was some interesting correlation between the property of the nucleotide and the property of the amino acid, but indeed you have noticed, Tamar, right? Tamar noticed that the correspondence was incomplete, imperfect. Indeed, it's imperfect, and this is indeed when you try to find statistical rules within biological data, it always comes with imperfections. So I don't know if it's an imperfection that we ignore and cells ignore, maybe we ignore and cells don't ignore, maybe it destines wrongly the mRNA to the wrong destination as a result of that. I think it's a good question. Hebrew, you want to see Hebrew? It's amazing. That's Hebrew. 2 things to be noticed. First, there is this bump here. It actually feels like an artifact to me. I can't understand it. But I checked, when do they cross? And that was in 73. Okay, so wars in the history of nations have a weird tendency to change relationships between question marks. English, yes. I knew it. All the questions are going to be on that and not on motif. No, you have to ask me a question about the regulatory motif, and then I'll show you English. Yeah, over there. I'll show you English in a minute. Yeah, you.
Do you have a candidate to read the signal?
Oh, good. Who reads the signal? I told you that we think, at least metaphorically or idealistically, not metaphorically, we think about a protein that would recognize that signal. It should be a protein that doesn't care, for the biologists among you, doesn't care about the reading frame, because as we said, the Y can be in any offset, as long as that's what we think. As long as this is in jumps of 3 nucleotides, it's only the ribosome that knows how to identify the reading frame. The rest of the entities in the cell, even in the cytoplasm, are not known to have that, except for some interesting exceptions in splicing, perhaps. So, but it's a long way to say that we don't know. But in Jeff Gerst's lab, they are trying now to identify these proteins. So what they do is that they put within a cell something that has this very strong pyrimidine content, and something that has exactly the opposite, the R content, the purine content. And you pull down the proteins that are binding each one of them, then you send them to mass spectrometry to identify the chemical nature of the binding proteins, and there are some interesting hints. That's what I can tell you at the moment. The previous question was who binds the motif? So the story that unfolds from this very small sample that we did. Now we are doing it on a much larger scale of many, many alternative motifs. Each cell is getting a different version, a different policy of what to produce and what to secrete, and they are forming a community of various degrees of cheating. This was a simple experiment in which you either a very strong cooperator, a wild type, or a very weak cooperator. When we grow them alone, the cheater, of course, doesn't succeed, especially when we simulate what would be the result of their growing together, but in a naive simulation that does not take into account the cooperation and donation among them, it is predicted that the cheater would die out. But when we actually grow them alone, and that was the other slide that I didn't show, the cheater is actually doing very well. It is together, yeah. When we actually grow them together, a co-culture of the 3 strains together, the cheater is actually quite happy. It is sustained in the population, presumably because it benefits from others. Whether something similar to what Adi showed us, that it could be later on disappearing from the population, or perhaps lead due to the tragedy of the commons to the demise of the entire population, we are actually looking at it now. But it doesn't seem that the population has collapsed. Yeah, I hope, if one is allowed to hope what to expect from an experiment, I hope it would not be the result. We did some theoretical predictions, some mathematical modeling that suggested that perhaps when there is a whole continuum of cooperation, not just a binary switch between cooperate or not, perhaps there are some regimes of the space of parameters that allow coexistence of the various versions, but indeed we'll see that very, very soon. And towards the very end, I'll just show you how English looks like. Thank you very much.